Psychology and neuroscience research studies use living beings as subject matter, so experiments involve social interactions between researchers and participants. As far as we know, geochemists don't worry if the quartz in their rock sample knows it's being observed. Psychologists and neuroscientists do have to worry about this, since both human and non-human animals act very differently when they know they're being watched. Also, experimenters must be careful that their own biases don't alter behavior of their participants. We'll consider two types of expectancy effects: participant and experimenter. Anyone who has the capacity to expect something can be susceptible to expectancy effects. Participants in a laboratory setting, like in Milgram's obedience study, often behave in such a way to please the experimenter. The experimenters themselves can also influence participant behavior if their expectations lead them to treat participants differently. Lastly, if confederates are used as part of the research design, they too can influence behavior of participants because of their expectations. For this reason, the use of confederates often involves pre-recorded voices to standardize the confederates' behavior across participants. We'll consider the first two types of expectancy effect, starting with participant expectancy. Martin Orne's attempts to find a control condition for his hypnosis research led to the discovery that a laboratory setting prompted people to act as good participants and to persist with a futile and boring task for 5.5 hours. So the laboratory setting can influence behavior in powerful ways. But research doesn't have to happen entirely in a laboratory setting to alter behavior in this way. Placebo effects are amongst the best-known participant expectancy effects. This study compared two different antidepressant drugs, the selective serotonin reuptake inhibitors sertraline and citalopram, and used a placebo control for comparison. The researchers used self-report measures to assess depressive symptoms over time, like the Hamilton Depression Rating Scale shown here. The y-axis shows the average change from baseline for each of the three groups, so the downward trend indicates improvement, since higher scores mean more symptoms. Citalopram produced the quickest and largest reduction in symptoms, followed by sertraline. Even though placebo wasn't as effective at reducing symptoms, on average the participants in this group showed a 10-point reduction, which is meaningful. The researchers looked at the data in a different way to show the extent to which each group improved, shown in this graph on the right. The y-axis shows the percentage of participants whose Hamilton depression scores declined by 50% or more after the onset of treatment, taken as a measure of remission. Over half the participants in both the citalopram and sertraline groups showed a 50% reduction, but almost 40% of the placebo also showed this degree of improvement. So it seems like placebo is a very effective treatment for depression. Placebos don't always lead to improvements. It depends on what the participant expects to happen. This clever study tested the power of expectancy on psychomotor performance. They used a pursuit rotor task where participants track a moving red dot with a cursor. Participants were undergraduates from the University of Waterloo, recruited for what they were told was a study on the effect of alcohol or caffeine on a motor skill. Researchers randomly assigned participants to 5 groups, none of whom were given any actual drugs. The control group completed 12 trials of the pursuit rotor task, then another 12, and the average change in performance plotted on the y-axis. This group did not show much improvement over these trials, so there's not much of a practice effect here. After the first 12 practice trials, the other groups were given what they thought was caffeine or alcohol, but were given different information on what effect this placebo would have on performance. Some participants were told that caffeine or alcohol typically enhances performance on this task. The height of the bar shows the average improvement, So those that thought they had caffeine and expected it to help did show an increase in performance. Those that thought that they got alcohol that would enhance performance showed a small improvement, but not as large as the bogus caffeine group. Those participants who thought they got caffeine and expected it to impair performance showed a slight worsening, a powerful effect when compared to the bogus caffeine enhancement condition. Surprisingly, those given fake alcohol and the expectation that it would impair performance actually performed better. Alcohol is a weird drug. When asked afterwards, many participants in this group reported that they tried much harder to overcome the expected effects of alcohol, and with no alcohol present in their system, their efforts manifested as improvement. Again, none of these participants were given any drugs, so the change in performance between groups is driven instead by their expectations. One approach to deal with the strong effects of placebo conditions is to include an additional, totally untreated control condition. In Verdon's experiment, there was one experimental condition, the power balance bracelet, and two control conditions, a placebo bracelet with the hologram removed and no bracelet at all. Since the results for these control conditions didn't differ, they were combined. Each control group targets a particular issue. The untreated control condition tells us how much of an effect the treatment produces, while the placebo tells us how much of the treatment effect can be explained by participant expectancy. Placebo-type conditions are also used in animal research. The rats in this study probably had no expectations about the effects of cocaine, but drug delivery involves injections, which can cause a short-term increase in activity. With a control group given physiological saline, similar to contact lens solution, researchers can distinguish hyperactivity from the injection procedure from the hyperactivity caused by cocaine. Test yourself. Discuss how using multiple control groups can strengthen the internal validity of an experiment. The experimenter can have a powerful influence on behavior, even in laboratory animals. These figures show two stress measures, levels of circulating corticosterone on the left and the number of poops on the right, for mice tested by male or female experimenters, shown in blue and pink, respectively. As shown by the arrows, mice handled by a male or exposed to a t-shirt worn by a male had larger stress reactions than those handled by females. Not surprisingly, human experimenters can influence human participants as well. Experimenter bias refers to a situation where the researcher's expectations of the outcome of the study actually influences participants' responses. This is also called the observer expectancy effect or the Rosenthal effect, after University of California at Irvine professor Robert Rosenthal, who published several experiments demonstrating this. This is an example of a self-fulfilling prophecy, whereby an individual's expectations about an event changes their own and others' behavior in such a way to reinforce the initial prediction. If this happens to an experimenter in a research study, it can weaken the internal validity. Let's consider a couple of examples from Rosenthal's research. In one experiment, 12 male graduate students served as experimenters in a study rating the apparent success of people from photos. The grad student experimenters were randomly assigned to groups, who were told either to expect low or high ratings from participants. Then they tested undergraduate participants using strict instructions to only read what was written to explain the task. Then the participants were given 20 photos of people and asked to rate how successful they appeared on a -10 to a +10 rating scale. Even though the experimenter instructions were identical, those who were tested by an experimenter who was expecting low ratings actually did give lower average ratings than those whose experimenter expected higher ratings. So the experimenters must have been doing something to alter participant behavior. Rosenthal showed that experimenter bias can affect the behavior of non-human animals as well. In one now famous experiment, Rosenthal used 13 undergraduate students enrolled in a lab course. 12 of these students had no previous experience working with rats, and one student had previous experience as a research assistant. All students were required to train food-deprived rats on a simple T-maze to learn the location of food. The 12 rat-naive undergrads were randomly assigned to groups and told one of two lies: that the rats were maze bright and would do very well, or were maze dull and would do badly. The sole student with rat experience was told to train the rats well on the task. Then all experimenters tested lab rats on the T-maze 10 times per day for 5 days. There were a number of dependent measures taken from both the human participants and the animal subjects, but we'll focus on two that measure rat performance: the number correct and time to complete. On the left is the average number of correct responses per day. The experimenters who were told their rats were bright obtained higher average correct responses from their rats than those who were told the rats were dull. On the right shows the average time in minutes to make a correct response, and again, those who were told their rats were bright got faster times for correct responses than those who were told their rats were not so smart. Note, the best performance was obtained by the student research assistant, not surprising given they had previous experience with testing rats. Test yourself. What strategies can researchers use to minimize expectancy effects? These studies demonstrate how expectancy effects can occur in both human and non-human animal research. Expectancy can arise from multiple sources, which can be problematic for researchers if they don't know about them. Strategies to deal with expectancy effects can avoid these potential confounds.